Well, thank you, Rodrigo, for that kind introduction, and thank you to the organizers for inviting me out. It's a pleasure to come up to Vancouver. Recently have moved to Seattle, but this is my first time to come up to Vancouver. I'm actually coming back in 3 months for another event, but it'll be great to come and see a real beautiful part of the world. So it's been great to meet with a number of people, and so as I was preparing this talk, I was hoping that I could be in the spirit of what VanBug is about, and so I've tried to focus a little bit on methods, as I was told is sort of the focus here tonight. And so what I want to tell you about is just some of our work with integrating regulatory and metabolic networks. And so just as an overview, I don't want to go into any of the details here, but other than just to introduce the field of systems biology, which is in essence trying to take large amounts of biological data of various different kinds and try to take this and distill it down to something where we can come up with predictive network models. And so that's kind of the flavor of what I want to talk about today, which is just how do we take, in this case, data from two specific and different kind of networks and integrate them together. So in terms of thinking about predictive models for biology, I would group these very roughly into two broad categories. You could do it in other ways, but this is one way to think about it, which is that we build models either that are based on biochemical reaction networks, which is one half here that I'll talk about, and on the other, based on statistical inference. And so I'm going to talk about a method sort of spanning both of these areas. So on the side of biochemical reaction networks, typically what we're interested in here, and we do this a lot for metabolic networks, is to have reaction stoichiometry, so we know something about the chemistries that are happening. We do a lot with constraint-based modeling. My thesis advisor was Bernard Paulson at UCSD, and so that's sort of where my training comes out of as a first case. And so I'll talk a little bit more about this as we get into it. And then on the other side are statistical inference type approaches where we don't know a lot about the chemistry, but we can try to learn from data. And the last talk was sort of a beautiful example of how we can apply this very broadly, even into new areas that I didn't know that we were doing this kind of work in for salmon. So very interesting. And so one of the areas that I'm really interested in is sort of how do we build models that can bridge between these two different areas. So the biochemical reaction networks and the statistical inference networks speak very different languages, and it can be hard to kind of get them to interface. And so that's one of the things I want to talk about today. So just to give you an overview of what I'll talk about, I'm going to tell essentially three different stories, three different methods that I'll present. The first I'm going to talk about is just straight regulatory network inference. I'll talk about a new method that we have for this, assessing subsets of transcriptional regulators influencing expression, or asterisks. As you'll see, it's really based pretty heavily on some prior methods, but it led to some quite interesting results, I would say, in honeybee brains, and I'll talk about that in a minute. The second story that I'll tell you today will be about how to integrate these genome-scale regulatory networks with genome-scale metabolic networks and introduce a new approach that we have called Probabilistic Regulation of Metabolism, or PROM. And then finally, I'm going to talk about a very recent approach. This was developed in the last few weeks, so this is the first time I've ever talked about this, so we'll see how it goes and what the questions are, and hope to get good insights from people here. But this is sort of a fundamentally new approach, I would say, where we're trying to use a metabolic network to help us with regulatory network inference. And I'll kind of explain what I mean by this, but this is essentially taking a very different kind of data and integrating it in to try to solve a problem in a very different space. And so I'll talk about that. Okay, so the first story that I want to talk about is something that we did recently for reconstructing a transcriptional regulatory network for behavior. This is done in honeybees, and it's going to be done from brain tissue. So one question is how strong is the connection between gene expression and behavior? Right? Can we say anything about behavior by looking at the expression of genes? Right? A priori, the answer to that might not necessarily be yes. So we're very interested in, are there distinct neurogenomic states? So the transcriptome here in the brain we'll call a neurogenomic state here. But are there distinct neurogenomic states that underlie behaviors? What are the regulatory effects between transcription factors and these targets? Are they persistent across different behavioral states? And how are these neurogenomic states defined and maintained? Okay, so these are some of the kinds of questions that we might be interested in. So the model organism here, as I mentioned, is the honeybee. And so this was a collaboration with Gene Robinson at the University of Illinois. For those who don't know, Gene is a world expert on the behavior of honeybees, and in particular on the connection between genomics and social behavior in honeybees. And so he came to me a couple of years ago, having finished a 5-year center grant on this from the National Science Foundation, and said, well, we've done 24 experiments on this, and would you be interested in maybe looking at this from a systems point of view? And I said, well, 24 experiments is, kind of a start, but interesting. And he says, that's over 2,000 microarrays. And I said, oh, that's pretty serious. And so let me take a look at that. And so we started a collaboration. So here, what Gene has done, just to give a background, so this was published, a couple of different advances on this in Science by Gene in 2003 and 2008 that started to establish the notion that there were actually neurogenomic states that changed between certain phenotypes of bees. In this case, he was looking at an aggressive bee versus a normal bee, and it turns out that there are actually signatures or that you can identify at the gene expression level that will be predictive of whether or not a bee will become aggressive and sting you. This is actually quite interesting, and I'll come back to this later, because the time scale of change for gene expression is, of course, much longer than the time scale of a decision that happens in a neuron firing for the decision to sting you. So as I'll come back to a little bit later, this means that the gene expression signature that is there in some ways could be thought of as being indicative of the predisposition towards making a decision or not. And this is something that's really not worked out well at all at this point, which is how do those states actually relate or influence what's going to happen in terms of neuron firing. So this is an open question and one that I think is quite interesting. But we set out to do something a little different, which is to look at gene expression now very broadly across many different contexts in honeybees, and to essentially measure first, which is what Gene's lab did, measure gene expression over a wide range of behavioral states, integrate expression. So we're going to try to learn a predictive model to see if we can identify influences between transcription factors and putative target genes, evaluate this model under new conditions to see if predictive ability holds up, and then we'll analyze the properties of this network. So in this experiment, we ended up having 853 individual bees representing 48 different distinct behaviors. Okay, and so we had different kinds of bees. We had aggressive bees, right, that undergo, which you can stimulate physiologically via some alarm pheromone. You also have interesting evolutionary differences between, for example, African bees and European bees. African bees become, right, killer bees, as people have probably heard of, become very aggressive very easily, whereas European bees are fairly docile and hard to get to sting you. You have foragers that go out and look for food. You have nurses that take care of the young. You have bees that are aging at different levels of development, and you have soldiers. And so there's all kinds of different social contexts and social behaviors that are going on here. And we grouped these very broadly, or Gene, I should say, grouped these very broadly, because this is his area of expertise, into aggression, behavioral maturation, and foraging phenotypes. Okay, so what we set out to do, from kind of more of the bioinformatics point of view here, was to reconstruct a transcriptional regulatory network, or what I really like to refer more to as a meta-transcriptional regulatory network underlying behavior. I say meta because the data that we're looking at here is actually whole brains. So you're actually asking sort of a strange question in some way, which is the average expression of a transcription factor in a brain and somehow indicative of the average expression of a certain transcription factor? So we don't have spatial information here. In something I'm not talking about today, we've done a lot of analysis of spatial gene expression in mouse brains. So if people are interested in that, I'm happy to talk offline about that. But here we don't actually have spatial information. So what we did is to use the following approach, which borrows quite heavily off of a number of existing approaches. But basically, we take our input data, We then infer a skeletal network around a target gene. And for this, we just use Arachne, which is a mutual expression-based approach. We've tried a number of different ones, but that works as well as anything for this particular step. We then prune out indirect interactions. This is just using essentially looking at, this is a data processing inequality where you essentially look at if you have relationships between A and B, B and C, and A to C, you cut out whichever of the trio is the least predictive, since it can be predicted from the other two. And then once we do that, we then take that network and then we apply least angle regression to it. So in essence, we're trying to learn a quantitative model. So the goal here is to say, given the expression value of the transcription factors, can I predict the expression value of some number of genes in the brain? We then apply this in the training set and learn which of the genes we're able to predict well in the training set, lock down the model, and then we move into the test set, and then this establishes whether or not we are able to predict under a new experimental condition the relationships between transcription factors and targets. We then quantify those interactions, and then in the end we end up with a genome scale predictive network. So this Lars aspect here, when we're learning the regression, is also meant to enforce additional parsimony here, in the sense that we lose also a lot of the interactions that come from Arachne. We prune it down, but we prune it down actually even further as we build the predictive model, and that's the reason for using Lars, is we tried to focus on just the strongest predictors possible. Yeah?
How do you know when you prune interactions that it's not a real interaction?
You don't know that for sure. So one of the things I want to make really clear here is you don't know for sure. So if we say what's known about transcriptional regulation in the honeybee brain coming into the study, the answer is almost nothing. So you don't have, so I will not claim, certainly, that this is highly mechanistic. I'll show you in a minute the extent to which we were able to test possible mechanisms and show you what the evidence for that is. So right now, though, I'd only claim that we were able to get good predictive accuracy, which I'll show. I'll show some evidence for mechanism, but you don't know for sure. And there's another issue with the way this is done as well, which is that I mentioned that we do everything sort of from the standpoint of parsimony. And we do that sort of by necessity, because you're really worried in this kind of set of overfitting. And so we're really trying to pick out things that we think are the most believable, knowing that we're missing a lot. And in fact, we think we only know even 1/3 of the transcription factors that are likely to exist in the honeybee. And the reason, I'll make just a quick point about parsimony. So here we do parsimony, I said, because we're forced into it. But in biology, I actually don't really, biology is not parsimonious in its core, right? If you look at like the work that Eric Davidson has done, right, at Caltech on sea urchins, which is really beautiful and where they've worked from the bottom up, one of the aspects, that I advocate sort of, broadly on transcriptional approaches is, most of those really verified functionalities are not parsimonious. So for example, right, the double repressor gate, right, where you have A represses B, B represses C, is one of the most common motifs that are observed in gene regulatory networks. All parsimonious approaches miss that, because everything that is based on parsimony would say, A activates C, right? So there is a serious issue that has to be addressed, and I'll throw that out here because I think it's something the community really has to think about. Now, most methods, of course, we still do enforce parsimony, and the reason for that is that there are many ways. There are many ways to build a parsimonious network. There are many more ways to do a non-parsimonious network, and so I don't think that there are good ways right now to really get at that. I'm going to come back to this issue at the end and talk about some of the ways that we're trying to address this issue. So this probably sounds a little bit confusing because I'm sort of advocating for something that's a little different from what we actually did, but I guess I find myself almost continuously in that position, in the sense that, right, we can always imagine things that are better. But anyway, So let's, anyway, I hope that answers the question. So here's what we ended up doing, though. But in this case, we did try to just focus on those things that we thought were the most predictive, since this is the first time that this has ever been done for a brain, as far as I know. So here, so we built this predictive genome scale model of a meta, transcriptional regulatory network for a brain. And what we found was we were able to predict the expression of over 2,000 genes with a correlation of 0.87 in the test set between predictions and experiment, which came out actually quite a bit better than I thought it would. In fact, when I first asked Sriram to do this, I said, look, this is going to fail miserably, and then when it does, then we will then pick some reasonable subset of this to look at in detail. And he ran it, and we looked at it, and it ended up coming out pretty well. And so we thought that was quite interesting. So just to give you a sense for what these predictions look like, this is just a small subset. So there are predictions like this. There are over 2,000 of these. These are 4 representative examples. And when I say representative example, I actually mean representative example, not the 4 best that we've seen. This was chosen at random by MATLAB one time, well, 2 times. One time we did it, but there was a repeat in there because we sampled with replacement. But when I would give this at talks, people would always complain about that because it happens much more likely at random than not possible. And so I did it one more time. So this is actually a representative sample of what happened. So not a cherry pick sample. So this gives you a reasonable idea of what happens. So here, what you can see is that, so the blue here is the experiment. You can see quite a bit noisier than the red, which is the prediction. But as you can see, it tracks it quite well. And then this is the training set here in white. And then the part here in green is the test set. And so you can see, again, this ability to pick up changes. And of the 2,000, the other interesting thing is that it's very enriched for differentially expressed genes. Like one of the things we thought is it just sort of predicting things that stay flat, but that's actually not true. Almost all of the genes that come out are the ones that are most relevant to the behaviors in the brain because they are the most differentially expressed. So this is the kind of predictions that we get, and so it shows that there are relationships that are predictable, that persist when we leave out phenotypes. We also we did this in one case in cross-validation, but we also did this in a more stringent case, which is we left out whole experiments at a time, right, where you have biological variants, you have it done on different days, you have all the sort of technical variants that comes in, and the biological variants, and we did exactly as well in that case. So there really was remarkably little overfitting here. So our root mean squared deviation in the training set was 0.34, and in the test set was 0.35. So we didn't lose too much. And I think that was based on the point that we did, that we were very parsimonious as we went through there, trying to focus on those interactions that we thought were the best, at the expense of no doubt missing many, many, many real interactions that could be predicted in more complex ways, with the caveat that you'd be much more prone to get into overfitting. Okay, so it was brought up about mechanism. So I don't claim that this network is fully mechanistic, certainly, and I claim only its predictive ability, but we also did want to get a sanity check on, okay, does this seem to make sense, these sorts of interactions that we were getting? So one interesting thing is that we created modules, and a module here just means all the targets of any particular transcription factor, and asked the question, could we identify a cis-regulatory motif in the upstream regions of each of these genes that was enriched? In other words, was there a motif that appeared within this set much more likely, much more often than you would expect at random? And the answer to that question was yes in 108 of the 190 modules. So we got really high enrichment for there being a cis-regulatory motif, showing that there was a possible unified binding that the transcription factor could have in the module. So we were quite happy with that. The other aspect was we looked at the modules to see if they were enriched for specific biological processes, and that was true in most of the cases, 87%. So there did seem to be some coherence to what was coming out. The other thing we did, and again, we don't have any of this data directly in the honeybee. In fact, when we got the reviews back from Science on this paper, which it wasn't accepted, and basically what they wanted was a lot of confirmatory evidence, right, for the interactions, which is perfectly a reasonable thing to ask for. But we estimated that the experimental work to answer the reviews would be over 5 years, and so anyway, so we sent it to PNAS, which is where it was published. And you can read all the details there if you're interested. So anyway, so the enrichment for direct physical interaction, since there is none in the honeybee, but there is in Drosophila, which is the closest really well-studied organism. And so what we did to just give ourselves another sort of sanity check was to say, in cases where our TRN predicted an interaction, and there was an ortholog of both the transcription factor and the target in Drosophila, were we likely to find that it had been characterized as having a direct physical interaction, say by ChIP-ChIP or ChIP-seq? And when we did that, compared to taking randomized interactions, we ended up with a p-value less than 10 to the minus 15. So there was definitely enrichment in the sense that these were much more likely to have physical binding orthologs in Drosophila. So in terms of mechanism, again, I don't make any really strong claims on that front, but we did see that a number of these were conserved in Drosophila, and so that was a good thing to see. Okay, so take-home messages from the first. So what we did here essentially was to build a quantitative model of gene expression relationships in a brain. Much to our surprise, these tended to have quite a bit of coherence and tended to be quite predictable. So this suggested, and remember, these are all simple linear models. There's a lot more work that could be done in order to find the nonlinear models. We didn't do it in the first pass, mostly because we were worried about getting into overfitting. The relationship between brain gene expression and behavior thus was stronger, more predictable than we'd previously thought. And then I brought up this point a little earlier, which is that there is sort of a sense of a kind of memory in these states, not like memory like we think about it, but just that if all you knew about the bees was gene expression, you could predict some aspects of their behavior. And I mentioned the case of aggression, right, where you're having something that's happening on a fast time scale in the neuron. There's some sort of predisposition in that state that is reflected. And that's true not just evolutionarily, but also at the level of phenotype, because you can give alarm pheromone, changes the gene expression, and you could tell that they had been exposed to this and that they were actually ready to sting or to attack. Okay, and I'll go on from there. Okay, so anyway, so that's an introduction to inference of regulatory networks. Some of you will be no doubt very familiar with that. And so, but what my lab does a lot of is integrating with metabolism. Okay, and so what we want to do here is to look at integrating a transcriptional regulatory network with a metabolic network into a unified model. Okay, and this will be this is work also by Sriram Chandrasekaran, who also did the previous study. I've been joking with him. It was mentioned before that I have a lab of about 25 grad students and postdocs. Everything I'm going to tell you today is from one of them. So it's all Sriram today, because I thought it made a coherent methods story. So anyway, you should hire him someday. He's really good. Anyway, so this was from his PNAS paper, a different one from 2010. Okay, so when we get into metabolism, I just want to introduce briefly to those who aren't aware the notion of flux balance models or constraint-based models. And so in a constraint-based model, essentially what we have is we have a metabolic network, and it could be another kind of network, but any network for which you can write down chemistry, like you can for metabolism. And essentially what you do is you're going to analyze this typically at steady state. So you're going to do something really easy, which is a flux balance, which means that for every metabolite, however much of it is being made is the same as what's being consumed. In equals out around every metabolite. Okay? So that's a flux balance. When you do that, that sets up a set of linear equations. This defines a hyperplane. Okay? So there's some subspace where these are true, and then you cap that subspace by setting Vmaxes and Vmins on every reaction. Right? This caps a space, and when you do this, you end up with a solution space. If this cone is sort of the iconic way that this is shown, the cone happens, though, only if you decouple all reversible reactions into a forward and reverse direction. So if you force everything to be positive, which is done for some of these kinds of analyses, this forms this convex cone, and this has sort of become a quite famous topic from Bernard Paulson, that you can then analyze solution spaces. So most of the time when we're thinking about models in physics or chemistry, you're trying to solve for the solution, right? That's not what you're doing in constraint-based modeling. You're solving for a range of possible solutions. So philosophically, what you're trying to do is you want to go as far as the data will take you, but not farther, okay? And so once you define these solution spaces, what's shown here is that you have a whole host of different analysis methods that you can apply. This is from my review paper in 2004. There's been a lot of development also since then, but just to give you a sense that there are many, many different methods. I'm going to tell you about just one of them, but there's a whole world of analysis that you can do in this space. Okay. So let's talk a little bit about interactions between metabolic and regulatory networks. So there's all kinds of reasons that you might want to study these together. Okay. There's the obvious ones, which is that regulatory networks are at controlled gene expression. Genes that are expressed become the enzymes that catalyze metabolic reactions. The metabolic network creates the building blocks, right, the nucleotides for the transcriptional machinery. There are metabolites that actually can influence transcriptional regulators. And so there's a lot of biology here that's interesting to think about these things together. And these are not modeled very well in terms of the complexity that really exists in cells. And even what I'll tell you today, I think, is an important step along the way, but there's a long, long way to go. So the state of the art when we set out to do this particular project, I would say, is regulatory flux balance analysis. This was done in Bernard Paulson's group. Marcus Covert was the first author. Marcus, of course, now a professor at Stanford. And so they built a model for integrating regulation and metabolism. It was motivated by data limitations, so it's kept to be quite a simple approach. It has regulatory networks represented by Boolean rules. Okay, and so in essence, regulatory network determines what genes are on or off, and then in the metabolic network, reactions are either present or not, and then you compute flux balance, okay, like I just described. The rules were taken for literature curation, so this was about a year of effort for Marcus as he worked through various different papers, putting this together, putting together networks under different environmental conditions, and in essence came up with a really beautiful model at that time and published in Nature in 2004. Okay, but there's a few issues with this that we want to make better, and the field was actually sort of stuck on this approach for quite a long time. And so we wanted to try to do something about that, and so we set out to build a method which ended up being a Probabilistic Regulation of Metabolism, or PROM for short. And what we set out to do is, first, we wanted to come up with an approach that's automated, right? So for every species, we don't want to spend a year combing through literature. For many species, that literature, of course, doesn't exist, so we need some automated way to build the network. We wanted it to be comprehensive from high-throughput data. And then one of the changes that I made in here was, rather than trying to think about this as a Boolean network where it's just going to be on or off, we switched to think about it more in terms of probabilistic Boolean. And it's not formally a probabilistic Boolean network, but that was sort of the way that we thought about it. And the reason that this is going to be important is that it means we don't have to have just 100% on or completely off. We can now have every range in between. Okay, so we can have a quantitative model, and we were hoping that this would improve accuracy. So in terms of the PROM model, we set up, it's just based on probabilities. So whereas the old model had, if transcription factor B is expressed, then enzyme A will be present, we now have a probability, which is just what is the probability that an enzyme will be present given that, say, a certain transcription factor is expressed. Okay, so you're still going to binarize the data and Booleanize and turn it into a Boolean. You do that because there's no really strong correlation between transcriptome and flux. So it's not like if the transcript goes up that the flux necessarily has to go up. That's not true. But we're just going to assign probabilities here. Okay, so then what happens is we constrain fluxes using these probabilities. So we have a transcription factor, right? We learn these probabilities from microarray data. Okay, so we're just learning a lot of taking microarray data, and we also take the structure of the network from interaction data. And so we're just going to learn the probabilities on top of it. And then you adjust the flux bounds for every reaction based on adjusting the Vmax based on the probability. So we say either it can be there or it's not, and then by that probability we adjust it and then recompute the optimal flux state. So this gives a way for the regulatory network to influence the metabolic network. So it's based on a constraint-based metabolic model, as I mentioned. This is just the math behind, the real simple math behind a flux balance analysis. You maximize some objective function. Usually this represents growth of the cell, subject to S times V equals 0. Right, which is just your flux balance, mass balance, and you have it between bounds, and there you are. And then what PROM adds to that is you add additional constraints based on regulation, and you then impose another minimization here, which is this here. So this minimization of kappa times alpha plus kappa times beta is essentially a penalty function. So you're going to establish soft constraints based on gene expression, and then say that the fluxes should represent as well as possible these changes in Vmaxes that we make based on gene expression. So you can go outside the bounds, but you incur a penalty for doing so. So it gives you the closest solution that you can get. Okay? And that's what's enforced by these equations here. Okay, so to build for E. coli, and we've actually done this for a number of different organisms, but in the interest of time, I'm only going to talk about E. coli so that I can get to the last and newest part. And so at E. coli, we used as a basis the metabolic model from Adam Feist in Bernard Paulson's group. It has over 2,300 metabolic reactions. We get regulatory interactions from Regulon DB, over 1,700, and we were able to validate against 1,800 different growth phenotypes. And so we compared the accuracy here of the E. coli model to that of the Nature paper that with Marcus Covert and Bernard Paulson. And in essence, what we got was fairly comparable accuracy. PROM gets 85%, RFBA gets 81%. So we were able to see an improvement, mostly because it picks up a lot better this ILV-Y than was before, but you can see other conditions in which it does a lot better. And so it sort of starts filling in. So we got it to be a little bit more accurate. In the example I'm not showing here for yeast, it actually jumps up quite a lot, but this was the primary one that we did first. And so here, when we look at increased comprehensiveness to the previous RFBA model, so it is somewhat more accurate, not massively more. But where we think we get a big win here is that it incorporates a lot more interactions. So there's many more predictions being made. So it's at a higher percentage, but yet it's much more global and comprehensive. We have, of course, more regulated metabolic genes and a few more transcription factors. The other, of course, big win is that this is learned in an automated approach from high throughput data, not reconstructed manually. So it says you can get it something automated. It's actually more accurate than what came before, and it's more comprehensive. And so that's what we see as the usefulness of the approach. And then you can also test it against, for example, predicting quantitative growth rates versus experiments. And you can see sort of the match here that's shown there. And then the details is in the PNAS paper. Okay, so to sum up part 2, so PROM is a new approach for integrating transcriptional regulatory network with metabolism. Compared it to E. coli, not to yeast. I took that out in the interest of time, but it improved accuracy, was much more comprehensive than the old models, as I mentioned. And it's also the first time, really, that there's been a model built that's genome scale for both metabolic and regulatory networks, or at least that's comprehensive in terms of making a prediction across all of the genes. I also took out tuberculosis, but we did build the first genome scale integrated model for tuberculosis to show that it could be done in a lesser characterized organism. Anyway, so that's our the PROM method. Okay, so what I want to talk about now is something that we've just been developing very, very recently. So these are, like I said, the first time that I will talk about this. So this will be fun. And then and will be interesting to see what people might think about this. So what we're trying to do now is that, so PROM takes as a given a regulatory structure and a metabolic network, and essentially it creates a link. But what we're really interested in is thinking about how we can learn in a unified way both a regulatory and a metabolic network using combined statistical and biochemical approaches. Okay, so this is sort of where we're trying to get to, and what I'm showing you today just sort of steps along that road. And so, so here, in essence, what we want to do is now I'm going to still keep the metabolic network fixed for now. And work that I'm not talking about today, we're building quite an elaborate program for learning metabolic networks. So there is that ongoing, but I won't talk about that today. But in this step, what we're going to do is we're going to say that the metabolic network is fixed. But if we know something about the metabolic network, does this help us with a regulatory network? This gets back to some of what I was introducing in the first project when we were doing statistical learning, and we started the conversation about parsimony and how there is really concern about, how many of these interactions are real. And so one of the questions is, if there are cases where we know something really solid about biochemistry, can this help us with statistical inference where we're really not as solid? Is there information there that we can exploit in order to help statistical inference by grounding it in biochemistry? Okay, so this is one of the things that we're really interested in. So the way I think about this is there a biochemical basis for network inference? Can it help? As I mentioned, the inverse problem of inferring networks is notoriously challenging, and in general not possible to solve completely, right? Usually provably so. And extremely many networks are possible when you go through inference, and overlap with mechanism can often be minimal, okay, depending on the context and the situation. And so can we make that better? So a question, because to my knowledge no one has ever done this, and so why would we link a regulatory network to regulatory network inference to metabolism? So one of the things that we get is it links expression changes with larger phenotypes via metabolism. What this means is that if you have a gene regulatory network and you predict expression, it's pretty hard to then just look at that expression and say, oh, the cell is growing faster, or the cell is growing slower, or it's going to secrete more butanol, or it's hard to say that. By linking it to metabolism, you can now answer those kind of questions. Well, what does this transcriptome actually mean in terms of certain phenotypes, right, that you get from metabolism? So it ties you into a whole new kind of data that you can think about. Okay, I'm going to talk about that. Second, right, as I mentioned, it just gives us a biochemical framework that's solid that we can link to. And then the PROM networks, the genesis of this idea was I had another student, Bojena Caballero, who was working on this, and what we really wanted to do was to get away from PROM needing to have this sort of lots of measurements of chip and chip-seq. We wanted to just infer everything, and we're still trying to do that. But when we took those inferred networks, PROM would fail. But when we took networks that were based on direct interactions or physical measurements, they would do really well. So what this meant was that the metabolic network could somehow tell the difference between the two. And we said, well, is this something that we can exploit? This is interesting. And so we came up with an approach which is still in progress, really, which we call Gemini, which is gene expression and metabolism integrated for network inference. Okay, so we're going to try to use the metabolic network to help. So in essence, what we have is we have transcription factors. Okay, we're going to use YEASTRACT, right, the database for yeast interactions. So we have all kinds of interactions here, and we have a certain subset which are the gold standard set. Okay, so these in dashed lines represent putative interactions, and the ones in gold here are their direct interaction set, which have been all experimentally verified, that there's an interaction, that there's a regulatory effect, and the mechanism is known. So these are the gold standard, the best that we know of things that really happen in yeast. And then what we'll do is we'll connect each of these putative targets to enzymes that are then going to have an effect. This is using PROM on the metabolic network. Okay, so we're going to say, if these interactions are true, what is the effect via the metabolic network on, for example, growth rates or knockout, or I guess in essence what we're really looking at here is knockout lethality. And what we're going to do first is something really simple. If an interaction causes us to make false predictions at the level of metabolism for when we're going to make a lethal or non-lethal prediction, we will discard it. And what we want to see is, does this throw away gold standard true reactions much less frequently than it does other reactions? Do we get an enrichment, basically? So this is sort of our first question. Is there information there? Once we come through, then we'll get a refined network. You can see this here. What we're hoping for is we'll get rid of these red false ones and leave us with an enriched version of the gold standard set, and then come up with a new integrated regulatory metabolic network for genome scale networks, which we hope will then be more accurate. Okay? All right. So this is how it comes out. So again, this is just the very first experiment. So we take yeast extract. So this is just looking at, we take 30,169, which are the total interactions that are there. Okay, so these are the total interactions. Oh, and I should say, this is not the total interactions of the database. This is the total number of interactions that has something to do with metabolism. Obviously, this approach doesn't help us with any kind of regulation that is not related to metabolism. All right, so we take this, and it turns out that there are 4,722 direct interactions. This is our gold standard set. And so when we apply PROM to it and we just eliminate things that make false predictions, so we get rid of 8,000 interactions, and we lose a little under 800 of the gold standard. So if we just ask, do we get an enrichment there? Is it more likely to kick out things that are false, or things that are unproven, than things that are really gold standard? The p-value there is 1 times 10 to the minus 12. So the answer is yes. It's not overwhelmingly so, certainly, but you get some enrichment. So this we thought was exciting as a first step, because it said, okay, there is information there. There's a lot of refinement to really get this better, but there is something that is potentially helpful. So the other thing that we wanted to look at was, okay, well, these are all done by ChIP or ChIP-seq. Like, there's a little more high confidence here that some of these things really are true, even though they haven't been established as gold standard yet. So what we also wanted to do was to look at, I'm slightly ahead of myself there. So one thing that we wanted to look at here was, okay, we got this enrichment, but what if we took the same number that we get and we just ranked them all by which have the best mutual information, or which are the best correlation? Is that as good as using a metabolic network? And this is what we got. So mutual information, if you just take all the best connections based on mutual information from gene expression. So now this is the approach that most people use. Right? You take interaction data, and then you look at gene expression changes, and you see if they share mutual information. And if they do, then you say, okay, this looks to be something real, and this other thing looks to be not. Right? This is the most used approach. This has a p-value of 0.002. Okay, so that doesn't work so well. And people in the field sort of appreciate this, right? This is a common thing. It was certainly a reasonable thing to try. It does have a p-value. I mean, it does work to some degree, but it's very minor. In this case, sort of surprisingly to me, correlation actually did better, right? So you kept 2,500 direct here. You get 2,600 with correlation, p-value of 10 to the minus 8. But with Gemini, the new metabolic network approach, you keep 3,944. So in other words, if you rank the interactions in yeast extract and try to say if I'm going to enrich for gold standard, you do much better by doing it based on the metabolic network than you do by correlation or mutual information, which is, of course, what everybody does. So this we thought was pretty interesting. Okay. And this high enrichment then, what we did to show was that, so the enrichment from Gemini was not just based on expression, but was actually due to metabolic network. And that's what we tested to try out these other approaches. Another interesting aspect was that the PROM network actually had more enrichments for known metabolic pathways, despite having fewer interactions. So even though you threw away tons of those interactions, if you then said, the targets, the putative targets that were left, are they cleaner in terms of, do they represent more specific metabolic functions? The answer to that question was yes, they do. And remember, the beginning set is already filtered to only involve metabolism, right? So everything here is related to metabolism, but the one where we filtered out a lot more, where we got rid of the 8,000 interactions, helped a lot. Okay. So then we got to the next question, which I jumped the gun on just slightly a couple minutes ago, which is, okay, we've done this now for interaction data, right? This is all just curating chip and chip-seq kind of information. Now, what does it do when you apply it to actually de novo network inference, right, of the kind that, that I showed for honeybees at the beginning? And so we decided to compare it against a widely used method, CLR, which was done by Jim Collins and Tim Gardner. Great method. And it's a mutual information-based method. And essentially what it does is it compares mutual information between a transcription factor gene pair to the background distribution of mutual information scores for all possible transcription factor gene pairs that include either the transcription factor or its target, right? So you're just looking at, you're trying to find interactions that stand out against this background distribution. Okay, the key point here, though, is it's just a method for network inference, mutual information based. Okay. So you'll remember for yeast extract, we got an enrichment with a p-value of 1 times e to the minus 12th. Here, we take CLR, but we embed within that network the direct gold standard interactions. Right? So we have, again, 30,000 total interactions. We have direct interactions here, the gold standard set 4,700. When we do the final versus CLR, we keep 4,137 of them. So that's a p-value of 1 of 10, less than 10 to the minus 33. So here it says, can the method identify mechanistic true interactions on a background of something that is inferred? And the answer is, yeah, that one you get a much better enrichment even than before. So the metabolic network, again, is able to tell the difference between mechanistic interactions and those that aren't. So this is getting back to the initial question, which is, how do we learn about mechanism, which has been such a hard thing to really do, from the mutual information correlation perspective. And what I'm arguing here is that at least in the subset where we have something that's really grounded in biochemistry, we can actually map it through that and get information about what is actually real. And again, we're a long way from getting something perfect. Right? There's a huge amount of work that should be done, but initial results are that yes, there is significant information here. Okay. All right. So that led us to the next question, which is, well, can we do this in an iterative fashion? So we see that there's information here. Can we learn a network, prune it, learn it, prune it, learn it, prune it, and get something that's better? Okay. So I'll show you this. This is the initial stage. And so what we have yeast extract, or yeast extract. So we have our physical interactions. We're going to refine a model using the PROM step, which is just to prune out interactions that we don't think are true. We're then going to add CLR in here to try to add in new interactions and then come up with a second refined model. Okay. So I'm going to try to explain this. This slide needs some more work to get it to be really clear, but I'll talk you through it. So here, consistency, what's meant by consistency here is when we take the regulatory network and we map it onto metabolism, how often is it correct in terms of its predicting lethal versus non-lethal for knockouts. And so here, what we get in terms of consistency, so with just yeast extract applied, we get 64%. Okay, and that's 274 transcription factors. The refined model, after we apply the first step of Gemini, then we get 88% correct, again for 274. So this is just a pruning step. And this is that pruning that I've talked about already twice, right, where we get the p-value of 10 to the minus 12. So we get better results here. And then the interesting thing is we then take CLR, we infer a bunch of new interactions. All right, so now we expand the model again, and then we prune it again using Gemini. And we end up with a refined model now that's getting 93% accuracy in terms of its matching with the knockout lethality phenotype, but it's now predicting it for 415 different transcription factors. And the enrichment for those gold standard set from here, right, so we did it here, p-value less than 10 to the minus 12th. Compared to that one, we get an additional now enrichment with a p-value of 10 to the minus 4th. So we're getting a little bit more enrichment again. So this is a process that we can now think about iterating through multiple times. So this is also interesting. So let me just talk you through this. This is what we have is Uwe Sauer measured the growth rates of quantitatively for 77 transcription factor knockouts in E. coli. And so what we said here is, what is the correlation between our prediction for the quantitative growth rate based for every transcription factor knockout and the truth? And if you just take yeast extract and you just believe it, every interaction is real, you get a correlation of 0.23 and a p-value of 0.02. So not terribly significant. Well, not significant. When you do the refined model attached to PROM, you now get, using the Gemini approach I described, now we get a correlation of 0.65. That's a p-value on the order of 10 to the minus 13th. We do CLR. We then refine the model again. We now get a correlation of 0.74 with the data, with a p-value of 1 times e to the minus 18th. And then for galactose, so this is now all the growth rates on a different media condition, galactose, we see the same thing, 0.23 goes to 0.4. Well, not really the same thing. It doesn't do that great at the end. So glucose really goes really well. Galactose we get better and then sort of top out. But basically it shows that you're able to go through sort of an iterative learning process, and that this might be an interesting path forward. And then you can do this sort of iteratively across different conditions. So you can learn on, say, one condition. So this just shows the accuracy that you get in terms of knockout lethality predictions, right? When you, if you have everything just at its base with yeast extract, and then as you take, if you train on aerobic and then apply it to the other conditions, you can see that you get an improvement. So by learning the model on aerobic and then testing it on the other conditions, on the other growth media, right, this improves from 0.97 to 0.99, from 0.85 to 0.87, from 0.88 to 0.9, from 0.86 to 8. And you can just see it sort of improves. Then when we train it across aerobic and anaerobic, again, we do better, and eventually you saturate. And so your precision here goes from 15% and increases, not massively, but up to 17.4%. So you get improvement as you train on one condition and test on another. Okay, so again, it's just approaches that we can start to think about how to make these better. If you go to a higher threshold, so in PROM I mentioned that there's a penalty function, and it has this parameter kappa, which in the PNAS paper we just set at 1, and that's been our default. If you make that more stringent, so if we ramp that up to 100 and we say, okay, you really, which is essentially just weighting the expression factor harder, if you go up to 100, what happens is you lose more direct interactions, but you lose a lot of the other interactions. Right, so here's the refined network, here's the original. You can see we lose a little bit of the gold standard, but we actually lose now a lot of the non-gold standard. And so if you look at the precision here, right, how often, what we make a call actually is a direct interaction, right? You just do the calculations shown here. You can go from having a precision of 15.7% up to 20%, which is a 27% increase. And so, again, very promising signs, I would say, that you actually can use these sort of biochemically detailed maps to help with the statistical inference. Okay, so take-home points to end. So, what I'm advocating here is that I think it's a very promising idea to ground statistical network inference with biochemical detail whenever possible. One of the areas where we can really do that is where we have statistically learned regulatory networks and biochemically detailed metabolic networks. Metabolic networks can be reconstructed very well these days. There's good methods for doing this. We can do it for many different organisms. There are automated approaches. Both the seed with Chris Henry have developed very nice approaches to doing this. We have an approach that we'll release later this year that's actually going to be integrated also with the seed, so it'll be added in there as well. So Gemini, this approach that I talked about, is, as far as I know, the first attempt to curate the inference of a regulatory network using metabolic network or using some sort of biochemical detail. And I really expect that this will be a significant growth area going forward. It's I've been very, very pleasantly surprised at sort of how well it's worked. You can tell by looking at the results that there is, there's definitely promise there. There's interest. There's still a long way to go to really get to where we want to get for really high accurate regulatory metabolic co-inference. But I think this is very promising. And so we've been quite excited about this. So where we're headed, as I mentioned, we're building integrated systems to combine regulatory and metabolic reconstructions, which we want to do in really powerful ways. And I've shown just the sort of the initial steps towards being able to do that. With that, let me just thank the people that actually do the work. So for this talk, I have to give lots of credit, obviously, to Sriram, who's the first author on both the PNAS papers for the B-brain and for the PROM method, and is also the one doing the Gemini approach. So today I decided to just kind of talk about one continuous thread in terms of methods, and so it's really his work. But I also want to acknowledge Seth Amit, who was involved in the B study. At that point, he was a graduate student in Gene Robinson's lab, really a brilliant guy. Did a lot of the deep biology there. He's now a postdoc in my group, luckily for me. So he's doing some great work there. And James Eddy, also a fantastically great grad student. I could have given a whole talk on his stuff and actually have before. But he was also involved in some of the analysis for the honeybee. So I want to acknowledge him. And then funding sources for this work, particularly NIH and NSF. And happy to take any questions. Yeah.
So you saw that the iterated analysis of metabolic network will tend to the inference of the regulatory networks. How about the other way?
Yeah, so we haven't tried the other way yet, but that's coming next. Exactly. So this is exactly what we did. So initially we had regulatory network inference and we had metabolic reconstruction package, which has taken us like 2 years to do. It's been very complicated. And then, PROM was, all right, we have these fixed, but can we learn a link? This is the next step, which is metabolism is fixed, learn regulation. And you're exactly right. The next step is, okay, let's let them both vary and learn them simultaneously. And that's actually where we're going. We're writing up a big proposal to the Department of Energy right now, where we're going to pitch this idea jointly with some really fantastic regulatory network inference approaches in Nitin Baliga's group.
The metabolic network can be well constructed. So with the noisy gene regulatory network, in what aspect you expect it?
Oh, how do I think it will help? It's harder. There's a reason we started the other way. Metabolism, I think, in general, is more solid. Well, the approach that we're building for automating metabolic network reconstruction is very probabilistic in nature. It essentially is a maximum likelihood solution across lots of probabilities. And so essentially, when you do metabolic networks, there are many cases where there's uncertainty. There's different ways you could fill in, and we have approaches now for trying to do that in a probabilistic way. But it could be that it's a very close call on a number of different fronts. So you could imagine that if you're also trying to learn a regulatory network structure, that you could try to identify pairs of transcription factors and target genes that appear together, say, very often across evolution. And it may be that by looking at by taking those sets together and then using a kind of a PROM-based approach and say, okay, how do we get something that's really optimized that will work? Now, there's a lot of hand-waving in that, because there's also a lot of worry for doing that, because once you start changing both sides, now your combinatorics are getting really high. And as we always worry about in this field, are we starting to just get into fitting noise or overfitting? I'm like constantly paranoid about overfitting in every one of our projects, and so I make my students do things like sit down at MATLAB and hit the RAND function and give me the answer instead of picking things. 'Cause I'm always worried about that. So it's a really important issue, but it hasn't been totally worked out yet. We're still trying to get there. But I think that it's going to be something that will be really important. The other thing I think is really useful is really taking advantage of evolution. So looking at co-occurrence patterns of genes together as they go across evolution. So this was David Eisenberg and Ed Marcotte and others have done a lot of interesting work on phylogenetic profiling and things like this. We had a method that we released in PLOS Computational Biology a couple months ago with something that we call correlags, which are genes whose co-occurrence pattern across evolution is highly correlated. And these are also, I think, another really interesting aspect to look at, where we can just try to identify different modules that really move in evolution as a set. This is also a way I think we can get away from parsimony in network inference, because I think what needs to happen is where we have certain modules that have really been worked out nicely in biology, we need to come up with good ways to come up with signatures for identifying, for example, the double repressive gates. What actually clues us into that those are actually there, because they'll never be found by parsimony, and yet we know from Eric Davidson's work that gene regulatory networks are filled with these things. So we need new approaches to do this, and so that's another area that I think is really important. So, yeah. Well.
Thank you for the talk. I have a question about the transcription and regulation network, and I'm glad you brought up modules, because I was wondering if you've seen modules of genes or sets of genes that set one behavior, things that one behavior apart.
We do. In fact, yeah, I shortened that slide set down about half from what I did last time. My sister was in the audience for the last one. She's like, I think people got that point by a long way before you were done. But so modules, we actually do find that. So we did a comparison and we said, we looked at the distribution of modules and asked the question, are transcription factors more likely to be centralized around the three major phenotypes, either aggression, maturation, or behavior measure. Yeah, that's right. And so, having my Rick Perry moment. And so, sorry, that would really have been good in the U.S., but not so great in Canada. Well, I won't get into it. So what we actually found was that when we looked at those broad categories, and we said, are there transcription factors that that regulate modules local, are they specific to a particular subset much more often than random? The answer was yes, they are. So what was interesting is that we actually did find these modules that were related to aggression, that were related to foraging, that were related to all these different subcategories. And so what was interesting about that is that those categories, we just impose them sort of from our human point of view. There's no reason to think that necessarily they represent underlying biology, but they actually do. And we were really surprised by that. In fact, even when we did just an unbiased clustering, those categories come out pretty strongly. And so what it meant was that the way that we think about that, there is actually a hierarchical structure to how these things are put together, and the modules do correspond to something that we might think of as biology.
And I have a follow-up question. There was a slide earlier where you have red line and blue line showing how blue line is closing
Yeah.
to the red line. So I was wondering, I actually don't know what the x-axis is, so I'll
Oh yeah, let me explain that. That's an excellent question. Let's see, get back there.
Actually, my question is, so assuming you're representing a number of genes, 600 genes or something like that.
No. No. So so I'll have to make this clear on that. So I appreciate that. So every plot here, this is one gene. And this is all of the experimental conditions that we're trained on. And these are the set that we're tested on. And so what you have then, so every point here represents the expression in 1B under one phenotypic condition. And so what you're looking at here is the, so this is for this particular gene. This is just its ID number in the honeybee, this AMO 1035. This just means across all of the bees under every condition, this is what its gene expression was measured as. And the red is what it was predicted as. Yes? No?
Is this number of bees? Or
Yeah, it's every,
number of cells? Okay.
yeah, It's a
okay,
bee.
So there are 4 genes that you're showing there.
I'm showing 4 genes.
So I was wondering.
And there are, right, 2,000 such plots, but I'm just showing 4 of them.
Okay.
Just,
So I'm wondering if you look at the modules of those gene sets together. So instead of one red line, it might be a more complex, higher dimension.
oh, we did not do that in terms of the prediction. We talked about doing that, and we still should do that. But we didn't do it. We decided that we wanted to just predict genes here, but you could certainly do, for example, biclustering and predict expression of modules. And in fact, that was the very first thing that we talked about doing. And I don't remember why we decided to go to single genes, but we did. So it would be a very, very valid, very good thing to do. And so, and really should be done, because it would be very interesting to look at that.
There was someone here.
Have you tried including microRNAs in the 3D model?
We have no data here for microRNAs. So I would love to do that. I think they're obviously really important. This was kind of a first step. And if we could get that data, I would very, very happily include it, because it probably has a big influence.
So do you think you could use your metabolic networks in the higher organisms? Do you have microRNAs?
I do think so. So we want to do this in humans very much. And so we're looking at building. So we have, and what another part that I ended up leaving out of the talk today for time. So we've now built draft metabolic networks using this an automated approach for 130 cell types and tissue types in the human body. And so we have gene expression data. We have in the lab, I mean, it's not hard to get this, but we have in the lab, right, all human transcriptomes in a database. And so, right, that are publicly available. And so that's exactly what we're trying to do right now, which is to take our 130 metabolic maps that we have, gene expression data that we have across all the tissues and different cell types, and essentially build draft models of this kind across everything. And we're trying to get, hoping to get, funding from the from the NIH to do this. And so this is, a major effort that we're initiating.
One of the later slides that I used,
Uh-huh.
started with the yeast and then used the PROM to PROM interactions and then add with the CLR, add more interactions.
Mm-hmm.
I'm not sure if I fully understand. Would the added interactions from the CLR, would they be a subset? No, they could be any.
They can be a super, they can be anything. So that's the whole point of CLR, is just to try to give us a way forward. I don't think that that's necessarily a particularly great way to do it. It's sort of an easy way to do it because they have such a nice package already put together. But this is where, when I talked about the future direction where we want to co-learn regulatory and metabolic networks, that's one of the areas, which is, how can we, better make predictions of what might be new interactions. So like I said, we're just barely starting this.
So you're removing a lot of, by pruning, removing a lot of interactions.
We remove a lot.
And then you're adding, it's typically probably just a small fraction of new interactions that are added, or how do the numbers relate? How many are removed, how many are added?
Yeah, so we had, let's see, I'm going to come back to this.
This one.
Let's see.
Right, the first arrow.
Yeah, I'll have to check to be sure. I don't remember offhand exactly what the numbers are, but it is fairly substantial in the sense that we make predictions for a lot of new transcription factors. Because in this model, there's only 274 transcription factors. Here, there's 415. So we go up a lot in terms of, this is essentially global coverage, because you infer everything, something for every annotated transcription factor here, whereas you don't in the earlier ones. They have to have been measured physically. So there are quite a lot of new interactions that are included. I don't remember what the exact number is, but I mean, in terms of number of transcription factors, it's not quite double, but it's 30% or something.
There was just, I think, that one.
Yeah, but then.
Follow-up question. So I switched on very close to the earlier question. So are you concerned at all that, I mean, one of your validations is that you can extract the gold standard from CLR, right? But
Yes.
CLR is working so badly that CLR is working so badly that it has a distinct characteristic and interactions are not good enough to meet gold standard on average.
This is a fantastic question. This is a fantastic question because that is exactly what we went through. So we did CLR and we said, okay, do we get enrichment from CLR? And the answer is, well, there's nothing in there to find. There's very small fraction, very small. So it's trying to find a needle in a haystack. So what we found, which was kind of interesting, is by putting the direct interactions in there, because you see that we do actually still get benefit from doing that. Right? You can see that with the CLR we get 200 more interactions from the direct that actually stay in, so there is something there. But yes, the problem is, so that is why you get such good enrichment. It's because CLR really does lousy. And so you're exactly right. This is exactly what we went through because we said, okay, let's do it. And I remember pressing Shriram on this. I'm like, well, what are we getting in terms of fraction? He's like, well, there's no mechanism in there. So it's a great question because that's exactly why we ended up saying, well, we have to embed something real here. And so what we really want to try is to see if this Egrin approach from the Baliga lab is really better at picking up mechanism. If it's not, though, the real question is, how do we get methods for regulatory network inference that actually have good mechanism? And so that's one of the reasons I'm really excited about this because I'm like, okay, the metabolic network can tell the difference between mechanism to some degree. How do we use that so that we can now go forward and learn new mechanisms? And that is the real challenge. We haven't solved that at all at this point, but that's where we're trying to go, and that's what we're really excited about trying to do the next year, on this front. So it's a great question. Yeah, it's exactly right.
Okay, let's thank Dr. Pratt for his talk, and then we can talk about the results later on.